I hope you are doing well. I'm Dr. Hashemi, Professor of Medical Mycology in Tehran University. And in this course, we talk about mycotic disease. Do you know mycotic disease what mean? No? The infection that the etiologic agent is fungi. What is fungi? Did you study the PowerPoint that I sent you before? No? Okay. Uh, our lesson is medical mycology. Medical mycology. It is a name of microorganism, microscopic microorganism that cause some infection in human. Medical mycology is the study of mycosis. Mycosis means mycotic infection. The infection that cause is fungi, fungal element. Medical mycology is a study of mycosis in human body and their etiologic agent. Mycosis. What mean mycosis? Mycosis is disease that cause from fungi. Mycoses are the disease caused by fungi. Of the several thousands of species of fungi that you know, there are some species that lead to infection, about less than 300 species. What is fungi? I like somebody explain about fungi.
There is some organism like bacterias.
Yes?
It's related to another organ to get nutrition.
Yes, yes. And somebody complete the definition that my friend tell about? Mhm. 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 It showed that you read the PowerPoint, yes? Thank you. Okay, thank you. Fungi are a microorganism that is eukaryote. You know what means eukaryote? Yes? It is eukaryote. Have a true nucleus. Eukaryote means have a true nucleus. In structure, in morphology, fungi are larger than bacteria in morphology, in structure. They are heterotrophic. What is heterotrophic? Cannot photosynthesize. Yes. They are heterotrophic. They are they have not chlorophyll. Therefore, their living is related to another organism. Some of them are saprophytic. Saprophytic, that means exist and live in the other tissue, other instrument, other media. on dead tissue. Saprophytic fungi live on dead tissue. Sometimes, and some fungi living on organism. That name is parasitic fungi. Parasite. Most of fungi are obligatory aerobic. Okay, other than facultative anaerobic. The fungi for live for growth need two base elements. First, carbon. You can find carbon in which material? Sugar. Sugar. The base of sugar is carbon. It is a source, it is a main source for growth of fungi. The fungi need carbon as a energy source for metabolism and structure. Another element that fungi need is nitrogen source. You can find nitrogen in which element, in which material, in which media? Nitrogen, protein. Protein, the base element is nitrogen, azote. Therefore, we need two essential elements for growth of fungi. First, carbon. Second, nitrogen. But there is difference between the uh, the totally of carbon and nitrogen. Fungi usually need to media that is carbon rich, but it is not for nitrogen base. Nitrogen limit. Therefore, fungi for growth need a media carbon rich, nitrogen limit. It is different. Oh. For growth, fungi need some circumstance factor, like as temperature, like as temperature. Uh, the optimum temperature is 22 to 27, about 25 centigrade. Most of fungi grows at this temperature, about 25 centigrade degree. Another factor for growth of fungi is pH. pH. Usually, fungi need to some and less acidic pH between 5.6 to 7. It is a optimum pH for grass. Wet water need for grass for fungi. Humidity about 75 to 85%. This is some factor for growth of fungi in media in addition to yes, carbon and nitrogen. One of the most important of structure in fungi is a polysaccharide that exists in cell wall and fungal wall. That name is chitin. Chitin is very important. Chitin is very important. That uh, contribute in a structure of fungal wall. It is very important. Microorganisms usually have not chitin, but the fungi have chitin, and it is a main material in fungal wall. In a structure, we have three form, three morphology of fungi. First, mycelial form. Second, yeast form. And some fungi have two forms. That name is dimorphic. Dimorphic means the fungi that have two forms, mycelial form and yeast form. What is different between mycelial form and yeast form? Do you know? Yes, yes, yes. Yes. When we have a fungi that is yeast, you see in a structure unicellular, one cell. Okay, but when you talk about mycelial form or mycelial fungi, it is multiple cellular structure. Mycelial, multiple cellular structure is only one cell. Dimorphic fungi have two shapes, not only yeast form but also the mycelial form. When we have, yes, yes, another question that asks you is what is different between when we have fungi in mycelial form, when we have fungi in yeast form. Okay, we have a dimorphic fungi. Sometimes it show mycelial form. Sometimes it show yeast form. When? In which condition? In which condition we have mycelial form? In which condition we have yeast form? Yes, 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 yes. Okay, okay, thank you. Thank you. The dimorphic fungi are pathogen fungi. It is very important. The dimorphic fungi are pathogenic fungi. In soil, in outside the body, they are mycelial form. And when transferred to internal organ, transferred to human body, change to yeast form because they can tolerate the 7 centigrade degree of body and therefore lead to disease, lead to infection. It is important. The most of dimorphic fungi are pathogen for human body. Okay, when you see a mycelial form, you must use microscopic observation. Microscopic observation. In microscopic observation, you can see filament fungi. Mycelial show filament structure. Filament structure. This is a schematic shape of mycelial structure. Some mycelium have septate or transverse septa. This one, this one, this one. And some of them haven't any septum. And it is important for diagnosis. When we decide for detect some fungi we must have an microscopic observation. We must pay attention to symptom, is there or not. It is important because some fungi that having symptom are pathogen for human body. If you find a hive, a mycelium in a specimen like as blood, like as CSF, you can decide for mycotic infection. One of the most important and pathogenic disease in fungi is mucormycosis. Do you know or did you hear this name? In COVID-19, we have an infection. As name is black fungi. Black fungi. Did you hear the black fungi? No. It is an important infection in COVID-19 that lead to death in somebody. Mucormycosis is black fungi. I asked you a study about black fungi in COVID-19. It is important. The etiologic agent of this infection is mucormycosis, is mucoral structure. Mucoral structure or mucormycosis show non-septate mycelium. When we see non-septate mycelium under microscopic observation, we decide for mucormycosis. I will explain next in next class about mucormycosis, but I want to introduce the importance of seeing the non-septate mycelium. It is important for detection between septate mycelium, non-septate mycelium. It is a key for diagnosis between aspergillosis that lead to respiratory infection and mucormycosis that lead to brain infection. And we find differentiation by attention to septum and non-septum mycelium. Okay, mycelial form is filamentous form. You see in microscopic observation, filamentous form of fungi, they may septate or non-septate. Mycelium hive. Hive, a, a small, mycelium is hive, and large mycelium and large filament fungi is mycelium. Mycelium or hive may septate or non-septate structure. Half. In this picture, you can see the morphology of mycelial fungi. Mycelial fungi grows circular form. When you inoculate in media, after 1 week, 2 weeks, you can see the colony, the circular colony with many different color, green, yellow, red, black. You can find multiple color in one fungi or in all fungi. It shows circular form of colony with different color. For diagnosis, we use the color of colony. We use of color of the colony for diagnosis the fungi. For example, it is Aspergillus fumigatus. It is Aspergillus fumigatus. Why? Because it is a approximately blue colony. But for detect and identify exactly a colony, we must have microscopic observation. You must prepare a sample from this colony and examine with microscope. It is a shape of one fungi, another shape of the other fungi. There are many aspects, many shape of colony in a species and genus of all fungi. About yeast form of fungi. The colony is shaped to bacteria in morphology. The colony is similar to bacteria. But when you examine this colony by microscope, you can see a cell. You can see one cell that larger than bacteria. That larger than bacteria. This cell produce budding. This cell produce budding for grass, for multiplication, for produce blastospore. When you see blastospore, you can decide for yeast. The stem cell is one circular or ovoid shape and produce budding. The name of budding is blastospore. If you see, if you see the colony of some yeast in microscopic examination, you see pseudohive or pseudomycelium. What is pseudohive or pseudomycelium? Mycelium, we have mycelium, pseudomycelium. Yes, pseudomycelium means false mycelium. Or in Arabic, kazib. In some yeast, in 37 centigrade degree in human body, it grows, and the shape of uh, yeast change to mycelial form. Yeast form grows, produce germ tube. Germ tube is pre-hive shape and lead to hive. This hive is named pseudohive, hive kazib, false hive. In one infection, in fungal infection, we have candidiasis. In candidiasis, in body, the microorganism produce pseudohive or pseudomycelium, false mycelium. False mycelium. It is the microscopic form of this phenomenon. If you see pseudohive, and yeast in a specimen from human, you can decide for infection. If you see pseudohive and blastospore in some specimen, like as blood, like as CSF, like as sputum, you can decide for mycotic infection. That name is candidiasis. I will explain in next. Thermally dimorphic. This fungi that we speak about and talk about it is thermally dimorphic fungi. Means temperature important for change the form of yeast to mycelial form. Temperature is very important. Okay. In structure of fungi, we have some element. When you see with microscope, you can see some element. One of these elements was hive or mycelium. Another element, another fungal element is conidia or spore. It is synonym, conidia, spore. In some fungi, we use the word conidia, and the other we use the word spore. The shape of colony, the shape of conidia or shape of spore is important for diagnosis of some fungi. For example, in mucorale species, you can see this shape. Or in Aspergillus, another important fungi, you can see this shape. Spore and conidio produced by uh, phosphorylation reaction. Phosphorylation reaction. And these show a flower shape structure. Flower shape structure. This structure include some element that end product of it is conidia or spore. When the conidia produce free, the term is conidia. When the conidia produce in a sac, the term is spore or endospore.
What does it mean, sporulation reaction?
Some fungi for produce the spore need some uh, some stage, and this stage uh, makes some structure. Okay, based on temperature, based on the media, and the end of this stage, you can find the spore or conidia. We see in microscope examination in practical class. Okay, for classification of fungal element, fungal group, we have four group of true fungi. The method of this classification is uh, sexual structure. We have ascomycota, basidomycota, zygomycota, and deuteromycota. We have four group of fungi. Ascomycota, basidomycota, and zygomycota have sexual reproduction, but in deuteromycota, you haven't any sexual reproduction. Some fungi are ascomycete, like as dermatophyte. It is a important fungi that lead to skin disease. I will explain, I think, in next class. Or this fungi is Cryptococcus in a group of basidomycete, lead to brain infection, or mucoral body that I told you before, lead to mucormycosis with brain infection. And some species of fungi classify in Deuteromycota, like as Candida, that lead to candidiasis, or Malassezia that lead to skin disease. It is a name, Oscospore, Basidospore, Zygon. It's not important for you. In addition to sexual reproduction, we have asexual. That name is arthrospore, chlamydospore. Some fungi produce arthroconidae. If a mycelium breakdown from septum lead to arthroconidia, like as dermatophyte in skin. Dermatophyte produce arthroconidia in skin, and with microscopic observation, we can decide for this infection, dermatophytosis. When we see in a skin scrap in microscopic observation and see the arthroconidia, you can decide, you can diagnose this infection is dermatophytosis, another skin disease. Another method for uh, reproduction is produce of chlamydoconidia. In some fungi, it is important, like as candida. In candidiasis, in candida species, you can find some structure that name is chlamydoconidia. It is circular and large fungal element that uh, is a way of asexual reproduction. Another way of reproduction, asexual produce to spores is sporangiospore. Sporangiospore uh, is a way in mucoral species that lead to mucormycosis. These fungi having any septum, the mucoral species haven't septum in mycelium, and the asexual reproduction is sporangiospore. In summary, mycology is the study of fungi. Mycology is the study of fungi. Fungi are divided in two major groups called yeast and mold. A yeast is a unicellular budding fungi or fungus. A mold is multicellular, thread-like fungi, filamentous fungi. Fungi are usually slow growers, non-fastidious, and are prevalent in our everyday world. I send you with email a quiz. Did you receive it? No? May somebody call the Taro?